Good afternoon, everyone. Thank you for joining today's webinar. My name is Shannon McGrath, the Business Development Associate here at Avance Clinical, and I'll be acting as the moderator for today. The topic that we're going to cover is how ClinicReady by Avance Clinical delivers preclinical project management and scientific and regulatory advice to get biotechs into the clinic faster. I'll be handing over to Dr. Gabriel Kremidiotis, who will be chairing today's webinar.
Thank you, Shannon. I'm Gabriel Kremidiotis. I'm the Chief Scientific Officer for Avance Clinical. I've been with the company for 5 years, and prior to that, I've worked as a drug developer in the biotechnology setting for over 25 years, having done a lot of work in taking small molecules from early discovery all the way to completion of proof of concept phase 2 clinical trials. Thank you all for joining us for this webinar, and I wanted to also introduce to you two of our presenters today. Dr. Jörgen Mold, who joined Avance Clinical recently and will be talking to you about the ClinicReady by Avance services during this webinar. And we're also very thankful to Dr. Paul Wabnitz, who is our guest presenter today, in talking about ethics submissions here in Australia, some of the common problems that we've seen with some of the submissions, and honing a little bit more the discussion towards how biotechnology companies that want to do the first in human trials in Australia need to be thinking about submitting their preclinical packages and the content of those. So without further ado, I will pass on to Dr. Mold to talk to us about ClinicReady. Thank you.
Good afternoon. So my name is Jörgen Mold. I'm a scientific affairs specialist. I've recently joined Avance. I've been here for about 2 months now. I have about 25 years experience in drug discovery and clinical development. Most recently, I worked in medical affairs in neuroimmunology for a mid-sized pharmaceutical company. Prior to this, I worked for a biotech company in preclinical drug discovery, taking a number of CNS active drugs from the bench side up into early clinical trials. And then prior to that, I actually worked in academia in neuroscience. I'm going to be spending a few minutes today to talk about a new service that Avance is offering called ClinicReady by Avance. So on the first slide, you can see that ClinicReady is actually an extension of Avance's services into the preclinical space. And what we do here is actually to provide drug discovery advice. So this is a consultation and also project management service. And this really positions Avance as a translational CRO, really taking basic discovery all the way through into the clinic into phase 2 proof of concept. So what are the services that ClinicReady offer? And on this slide, you can see a number of services as part of the ClinicReady package. So firstly, we provide a program gap analysis with a particular emphasis on preclinical study plans. So really understanding, what it is that you want to do with your drug, what kind of indication you're going for, and what preclinical data that you might already have, and what really is required in order to get this drug into the clinic. We can provide therapeutic area advice in terms of competitor analysis and really helping you to understand how this product is going to differentiate from other products which are already in clinical trials or on the market. We can provide scientific advice in terms of the product profile, candidate selection, providing a risk-benefit analysis and also help with data interpretation from preclinical studies, project coordination and management. So in terms of your preclinical studies, we can assist with vendor selection, management of timelines and deliverables. And then finally, culminating in the preparation of an investigator brochure. So this is the writing up of all your preclinical data into a format that would be used for submission for clinical trials. In this respect, we can also assist with regulatory agency submission support. So it really comes down to what services that you need, and we can provide all or some of these services, really depending on what stage of the discovery process that you're at. So in terms of a typical project overview, this shows you a typical 3-year drug discovery plan, really starting from new hit identification and drug screening, lead identification optimization, involving synthetic chemistry and developing compounds through a structure-activity relationship, and then selection of a lead series. And then this also brings through into in vitro proof of concept, in vitro studies, moving compounds into in vivo proof of concept, pharmacology, and then at this stage also understanding some early in vivo toxicology to really establish what the safety window is of your lead candidates. Following this, there'd be a small scale-up once you've selected a preclinical candidate, and again, selection of these candidates would be based on a set of desirable parameters. This would then go into small scale-up and allow you to then move into your IND-enabling studies, firstly non-GLP studies. So this is some preliminary dose range finding studies. This is also another opportunity to look at more complex, sophisticated animal models of human disease. Following this, there'd be a larger scale-up of drug, and then looking at drug stability and maybe some formulation work before then moving into the actual pivotal GLP tox studies, which are required before first-in-man studies. And of course, these GLP tox studies are required to really help with estimation of your first dose in human. Now, following all of this, we can use all this information to be populating the Investigator brochure again, which is required for submission. So this slide really just shows an example of a recent drug discovery plan that we did for a client, and really outlines some of the key assays. And this is all the way from hit identification all the way through to the selection of your preclinical candidate. And it just shows that there's really a series of key assays that need to be performed. And part of our process and service is really identifying what the key assays are. I mean, there's a number of assays that would be nice to do, but they're obviously not on the critical path. And one of the key parts of designing an assay cascade is really knowing what the end product is. So you really need to start with a target product profile and then work backwards. And then just really design your assay cascade to understand how you can very rapidly identify analogues that are on target, any secondary screens you might need to confirm target activity, some early in vitro ADME studies, then getting an in vitro proof of concept and a suitable in vitro assay. This would be followed by tier 2 in vitro ADME studies, understanding the PK, and then moving into your in vivo animal model to get proof of concept. Now, the in vitro EC50 values that you get can actually help to tell you what kinds of exposures that you're going to need in your PD model. Following this, compounds would then be taken into some preliminary toxicology studies, and then with your lead compound, you do additional safety profiling. The whole point of this is to really be able to rapidly ascertain hits and develop a very rapid SAR, but at the same time identifying any safety liabilities early so that there's an opportunity to medchem these out if possible and identify alternate series if required. Now, as you actually move through this cascade, obviously the expense or the cost of studies increases, so the more that you can understand earlier, the better. Now, once you've selected your preclinical candidate, these would be typically taken into IND-enabling studies to then identify your clinical candidate, which is then ready to be submitted for the clinic. These are comprised of non-GLP and GLP activities. So initially, your preclinical candidate would be taken into non-GLP activities, including formulation and scale-up, assessment of purity. We'll also be conducting your dose range finding studies, and these would typically be performed in a rodent and a non-rodent species, so 2 mammalian species. And again, this provides a further opportunity to be examining efficacy in in a more sophisticated model of human disease. Now, whilst these activities, the efficacy models are placed here, it is also possible to continue these, run these studies actually in parallel with the phase 1 first-in-human studies. Compounds would then be progressed into your formal GLP tox studies. These include things like, obviously, understanding cardiac liability through hERG IC50 determination, looking at genetic toxicity and mutagenic potential, and then doing your standard safety respiratory CNS and CD tox, and then your formal toxicology studies, which again are in 2 mammalian species, 1 rodent and 1 non-rodent, and these would be then used to estimate the starting dose in your first-in-human studies. So as an overall, these are some of the types of studies that are required in the preclinical setting in order to support first-in-human studies. And this is a fairly standard sort of package around what you'd expect for a small molecule. But it doesn't really outline all of the studies, and of course with new types of therapies such as cell-based therapies and biotechnology-derived products, obviously there are other assays that would be included in this kind of cascade which are not written here, and obviously some of these types of studies probably wouldn't apply in those situations. But if you look at the assays that you normally need to provide, it'd be obviously primary pharmacology. Of course, these are non-GLP, so in vitro, cell-based assays, in vivo data, so your animal PD model, pharmacokinetic data. So this really understanding, how your compound is engaging with the target and really what the in vivo levels are to do that, and also to get an apparent safety window between in vivo efficacy and any tox effects. Then, of course, the safety pharmacology, so provided through in vitro screens, hERG inhibition, and then your CNS, respiratory, and cardiovascular system safety. Then that would actually move then into your toxicology studies, so your dose range finding studies, rats, and then also in a non-rodent species. Dogs are often used for this type of study. Then you need to really understand more about metabolism, so plasma protein binding in all species, understanding microsomal stability, and it would also be expected that you'd have a very full understanding of the metabolic pathway for elimination, at least in humans, and whether there's any major metabolites that also need to be investigated. And again, genotoxicity would also be assessed at this stage. So Avance Clinical Services really allow a seamless integration with development under an IND type of program. So really the way that we would suggest for this to be done would be to, once you've identified a drug candidate and done some obviously non-GLP exploratory talks, this is in vitro and in vivo, this would be the point where we could support a pre-IND meeting. The whole point of the pre-IND meeting is to really make sure that the pivotal tox studies that you're doing are the actual crucial studies. And of course, these are relatively expensive studies to do, and you want to make sure that you're not using animals unnecessarily. So having pre-IND meetings is a really good opportunity to make sure that you're on the right path to having the required information for first-in-man studies. At this point, we then recommend going into phase 1 studies in Australia. And the reason for this is that obviously, although new medicinal products are certainly highly regulated in Australia, the submission is different. So the submission would the package would typically submit it to a Human Research Ethics Committee. And basically this allows you to get some preliminary phase 1 data, which then would help to support your package for IND submission before then obtaining your phase 2 proof of concept. So another advantage of conducting your studies in Australia is that there's a very large research and development tax incentive, which really provides a strong financial incentive to including Australian CROs, clinical sites, and investigator networks from Australia in this, to really get into these proof of concept studies. And of course, with a lot of our clients, the idea here is to really get this sort of data to then provide, capital raising for more expensive later phase clinical trials, or to then license out the compound to a larger company. So I'm just going to hand over at this stage to my colleague, Dr. Gabriel Cremidiotis, who's actually now going to talk to you a little bit more about what happens after this and some of the requirements for getting into early phase clinical trials in Australia.
Thank you, Jorgen. Thank you for this really nice overview, and also some of the insight that you shared with us regarding the preclinical activities and requirements leading up to starting evaluation in clinical trials. So once we start talking about clinical trials in Australia, I think it is important for our listeners to get a sense of what the schemes are, what the paths for getting approval to evaluate the compounds in humans. There are in Australia clinical trials regulated by the TGA, the Therapeutic Goods Administration, and there are two schemes that allow getting approval to conduct evaluation of a compound in clinical trials. One is referred to as the notification system, or CTN, and the other one is the clinical trial exception system, or CTX. By far, the largest majority of trials get approval via the clinical trial notification system, which also offers a rapid pathway to getting approval, and I will tell you a little bit about that. The CTX system is more reserved for high-risk products, including gene therapy and class 4 biologicals, and so most of the trials that we do are actually CTN. Under the CTN system, the scientific and ethical merit of the application is reviewed by the institutional ethics, or a central IRB that can handle some of the submissions in Australia. And in terms of the involvement of the Therapeutic Goods Administration, which is our regulatory in Australia, it is just an online a notification form that we usually complete for our clients. And the way it works is to basically do the submission to the ethics committee while at the same time pre-filling the notification form. So when we have approval, we can move as quickly as possible to activating site and starting the trial. So one of the very common questions that we are asked by our clients is with regards to the documentation that needs to be submitted to the ethics committee for consideration of getting approval to conduct a trial. The most important documents that I've highlighted here are the study protocol, the investigator brochure that describes all the known data and workup up to that point for a particular product, and of course the participant information and consent forms. There are some other ancillary documents that I'm listing here, including any questionnaires, the CVs of the principal investigators, any advertisements that may be used as part of the accrual for the study, indemnity cover. There needs to be a certificate of currency reflecting that there is an insurance in place for the trial. And most of the trials here in Australia are listed in the Australian and New Zealand Clinical Trials Registry, referred to as ANZ-CTR, which is sort of the equivalent of the clinicaltrials.gov, if you like, that you would find for trials that are conducted in the United States. Our team here will help you with putting together all these documents, including offering you the ability to prepare a study protocol, advising you with designs of studies, particularly, we specialize particularly in phase 1 healthy volunteer, as well as patient and oncology studies. We are able to work with you in terms of preparing your investigator brochure, and we can do this at a very flexible way. We can prepare it from scratch for you, looking at your preclinical reports, or we can do a gap analysis on these documents for you before submission. I just wanted to concentrate a little bit more, given the subject matter of this discussion, on the preclinical data that an ethics committee here in Australia would like to see. All that gets captured in the investigator's brochure. In its essence, the investigator brochure here in Australia tends to be a more data-rich document, because it is the only document that the ethics committee will review in the first instance. Provision of any other preclinical reports or documents to the committee is only by request. And Dr. Paul Wabnitz will talk a little bit more in his part of the presentation about this. So in terms of the investigator brochure, preclinical pharmacology needs to be presented that supports the rationale for the target therapeutic indication. There needs to be a clear safety, toxicology, pharmacokinetic, and metabolism data that guide us as to the proposed dosing levels for the clinical trial, and also gives support for the treatment schedule that we're looking to use in clinical trials, at least in the first in human trial in the first instance. The toxicology studies that Jorgen mentioned earlier, the pivotal GLP toxicology studies in 2 species are extremely important in identifying the target organs and tissues, or target physiological processes that are likely to drive any adverse events for the particular product, and are also very important in terms of guiding the types of assessments that need to be carried out, and the type of monitoring that needs to be done, particularly during the first in human trial. And overall, all these parameters lead into the early establishment of a risk-benefit profile for the intended product before we start a human trial. All these need to be captured in the investigator brochure and clearly communicated to the ethics committee in a way that indicates interpretation of data across the different aspects of these evaluations. And I just wanted to just concentrate a little bit more on that by giving you some real-time examples of the types of tables that we include here at Avance Clinical in the investigator brochures that we prepare for our clients. What you can see in this slide, it looks like a complex table, but what you actually see is that we are driving a clear correlation across the proposed dose levels for the clinical trial, what those dose levels are in humans, and how these correlate to dose levels in preclinical type species that are used as part of the preclinical workup, while at the same time capturing the exposure levels, the systemic exposure levels of the product, which are really important in terms of interpreting the types of concentrations that we need to cover the receptor. And you can see here we are marking the dose levels where we are seeing efficacy. And as we work down this concentration path, if you like, we see increased concentrations of systemic exposure. And here on the right-hand side, we indicate some key studies and key events that occurred during those studies during the preclinical workup. And that gives the ethics committee a real, a single snapshot of the dose levels that we propose, how they correlate across the preclinical species and human, and how do they sit in terms of the preclinical evaluations that were done. And that makes their job much easier. It makes interpretation on our part and the client's part much easier in terms of what the toxicology findings are, which are also captured in a similar sort of holistic summary, single snapshot format. And you can see some examples of tables here where we are looking at the toxicology findings. Histopathology is captured as long as gross clinical findings across the species and across dose levels that were used for the preclinical workup, while at the same time correlating the systemic drug levels, information that came out of the toxicokinetic component of these GLP pivotal studies. And though all these sort of cross-correlation across studies is extremely important in defining and providing a good justification for the starting dose, as well as the dose range that is proposed for that first in human clinical trial. And it is really important to present this to the committee. It makes their job much easier. Dr. Paul Wabnitz, who will present right after me, will talk to you a little bit more about that. He's a member in ethics committees, and he can speak to the importance of capturing the data in a holistic type configuration like this, enabling better interpretation of the findings and better correlation as to the risk-benefit profile of the product before it's taken into evaluation in humans. So it is important, I think, for all of you out there to get a bit of an idea as to the timeframes that are involved in getting ethics approvals for first-in-human trials in Australia. We at Avance Clinical tend to work a lot with a central IRB, referred to as Bellberry Limited. They have 12 committees. They have several meetings per month, and they guarantee a really fast-track approval process. The process from a scientific and ethical review perspective is still extremely rigorous, and it is exactly to the same level as you would expect to find in other regulatory jurisdictions. However, given that the committee specializes in this type of activity and has a number of committees, they are able to provide fast-track approval arrangements. Usually at Avance, for our clients, we get approvals from submission to getting approval in about 5 to 6 weeks, on average about 5 weeks. I noted there that for COVID trials, we are getting considerably faster timeframes, 2 to 3 weeks, because Bellberry Limited are actually prioritizing these type of trials, and they guarantee expedited ethics review for such protocols. So for this type of studies, first-in-human healthy volunteer studies, we work a lot with a number, and some oncology trials, we work a lot with the dedicated phase 1 clinical trial units that are located here in Australia, and I'm listing some of those here. They are located in all the major states in Australia. We are at Avance based in South Australia. CMAX is also based here. It's sort of 10 minutes down the road from our headquarters. We do a lot of work with them, but also engage quite a number of the other phase 1 units. And the teams are very much streamlined to execute a first-in-human clinical trial in a very timely fashion and with high-quality data outputs. There is also support from third-party vendors in terms of the management of the handling of the investigational product. I'm listing some of the vendors, the third-party vendors that we work with. So we advise clients to contact us early, particularly when it comes to investigational product. There are certain aspects and facets of the investigational product that they need to be aware of, particularly when it comes to aspects of formulation and stability, compatibility, and they need to be in place prior to starting a trial. So at this point, I will pass on to Dr. Paul Wabnitz. Perhaps, Paul, you can thank you for joining us today. We really appreciate your time. perhaps you can give us a little bit of the background, your background, and then tell us a little bit more about these aspects of starting first-in-human clinical trials and some of the requirements for our ethics here. And particularly, it'd be nice to hear a little bit more about some of the gaps or some of the drawbacks that you've seen in some of the submissions over the years. Thank you.
Thank you very much, Gabriel. Good afternoon, everyone. My name is Paul Wabnitz. I'm a medical doctor. I'm also a scientist and have a background in drug development and early phase clinical trials. I'll just give a very quick overview of my background. So I started with a science degree and then completed a PhD in organic chemistry here at the University of Adelaide. Essentially, that was with frog peptides and proteins. We used to collect them from frogs and then analyze the proteins and peptides by mass spectrometry in HPLC and then test for activity. And I developed an interest in oncology at that time because a lot of the peptides were testing for either for antibacterial properties or anti-cancer. I was very fortunate during my PhD and was able to have a good project where I could publish a lot and was able to publish a first author publication in Nature, which attracted a bit of attention overseas. So I was offered to then have an industrial postdoc, which was a little bit different back then. Most people were into academic postdocs, but I chose an industrial postdoc at Park Davis in Ann Arbor and did a postdoc in drug discovery. And that lasted for about 6 months because at that time Pfizer bought Park Davis and I was offered a scientific job. So I took that on and spent then 6 years working in Pfizer research in Ann Arbor in a variety of roles, but mainly in pharmacokinetics and drug metabolism and really concentrating on inflammation oncology. So I've had hands-on experience with not only designing the lab experiments, but actually conducting them myself. Also had experience in toxicology, toxicokinetics, and in overall drug design and development, and then also in early clinical trials. Was able to be involved in 2 very, very large phase 1, phase 2 trials, and again had to conduct some formulation stability studies for some very complicated biological drugs. So it's a really good experience. I was really enjoying it. My family is all in Adelaide, and so I at some point had to make the decision to come back to Adelaide and for family reasons and moved back. I completed then my medical degree here in Adelaide. Actually, one thing I'll just also point out, while I was at Pfizer, I had the privilege of being sponsored by Pfizer to get some additional training outside of hours, and I was progressed through a MBA, which as a scientist was actually quite helpful, gave me a bit of insight as to the operational aspects of planning and corporate strategy. And so I, over 3.5 years, studied an MBA at the University of Michigan Business School in the evenings and weekends. And then after that, I completed a drug development certificate in regulatory affairs at Temple University in Pennsylvania, which was also very, very quite interesting and very helpful. So coming back to Adelaide, I completed my medical degree at Flinders, and then I went through, and in Australia, we get to pick our pathways. Pathways include surgery, physician training, radiology, or general practice. I picked physician training because I thought that was the most likely compatible with my background. And I've then gone through the training pathway through the Royal Australian College of Physicians in dual training in clinical pharmacology and also medical oncology. I've actually finished clinical pharmacology and am a clinical pharmacologist and currently advanced training in medical oncology. I've also have quite a lot of phase 1 experience and first in human experience, and this is really where my interest lies. So I have experience as a medical director at CMAX for almost 2 years. I then stepped down due to other work commitments. I still am involved at CMAX as an investigator, and I'm a principal investigator on a number of phase 1 trials, which I find quite advantageous because I get to see and troubleshoot some of the processes and decisions that are made as an ethics reviewer as well. Also work as a medical monitor in oncology, phase 1 and 2 trials, which I really enjoy. So the point that I like to highlight is I'm also a regulatory approver and reviewer for the local ethics committees. So I do a lot of the toxicology and clinical pharmacology reviews, predominantly in complex clinical trials in oncology, hematology, and immunology. And I'm a primary reviewer for the public sector local hospital network, which is the Royal Adelaide Hospital and also the Central Adelaide Health Network, and also privately for a national organisation that Gabriel's mentioned, Belbury. And I spend a fair bit of time, I almost call it a hobby, because I do that out of hours in my own time. Some of the reviews can take up to 15 hours. Next slide, please, Gabriel. So I just thought I'd highlight some of the common questions that often get asked or often gets brought up when companies from outside of Australia, especially from the US, ask what's the main difference between the American system and the Australian system. So I thought I'd just outline that very briefly. I know that Gabriel and Jorgen have touched on this as well. The regulatory requirements here in Australia are very closely aligned with other agencies, including the European agencies, the American agencies, and also the International Conference on Harmonisation. There's no particular guideline that we use, but we refer to these. And so when we get asked what then is the difference between phase 1 first in human approvals in Australia compared to overseas, such as the IND process, really the advantage is that it is much more streamlined, and you don't have to have an extensive regulatory document such as the IND. However, the regulatory standards are exactly the same. And so the way that I like to think about it is they're essentially the same, however, they're more simplified, and you also, the biggest component of the IND that's missing in the Australian process is the chemistry, manufacturing, and control in the Australian IND. So in Australia, that component is really just the bare bone basics that I might touch on in a short while. But besides that, everything else, the standards are exactly the same. The other advantage that I see is that it's much more rational based. What that means is a few years ago, everything was small molecules where we followed a very standardized process. Now what we're seeing in the last few years, very novel, very interesting, complex types of drugs, from vaccines to gel therapies, gene therapies and gel therapies, yes, oncolytic viruses and all sorts of really interesting technologies. And there's no guideline that applies to some of these. So you actually have to then just use your own common sense approach. And so a lot of that's rationale based. And I think that's one of the advantages of Australia. We're really innovative, and we can go through and problem solve with these novel therapies. And we find that, especially when there's a difficult technology in the US, sometimes by coming to Australia, having that problem worked through at a much earlier stage is very advantageous to the IND process, as Jorgen and Gabriel have also outlined. And also, not saying that other regulatory agencies are box checking, but when you stick to just the guideline, it really limits the novelty of some of the drug design. And really, we're moving towards rationalization, where we want to avoid doing tests that are unnecessary. We want to avoid using animals as much as possible, but at the same time, we never ever compromise safety. And I think that's what we do well here. We can make that rationalization so we have an efficient and effective process. And I think that's why our regulatory process, when it's done well, is very, very fast. So that being said, then what are the core components? And Gabriel's already touched on some of these, so I won't go into a lot of detail, but I break it down into just headings. And so to me, the way that I look at it is the proof of concept. What is it about your drug that shows us that it works? And usually there's a very simple model that shows the pharmacological effect within the relevant disease. Then there's the chemical properties. So I mentioned that the CMC section is usually not as prominent in the Australian system. However, it is still important to include the basic chemistry. I often, as an organic chemist, will look at the structure and have a bit of a think, even if it's a biological, what kind of action that drug's going to have just by looking at the structure. So by having it there, having all the basic chemical parameters, the pKa, the log, and the molecular weight, is really important information. Besides that, the other parts that's important to include in there is, obviously you don't need to include the whole manufacturing process, but just an indication that it's been done in a particular way that allows for quality control and consistency. And one thing that we find is quite important that is often missed is the stability, just a very simple stability study. And I'll touch on that with common mistakes that I'll talk about in a short while. So then the pharmacokinetics, as Gabriel mentioned, it really is there to support the dose. Now, as I mentioned, I'm also a principal investigator, so I get to see what it's like to do the trial in real life with real people. So I use those values really to help me design at what point do I expect there to be a drug effect. And so with pharmacokinetics, you're looking at maximum time to exposure. So if it's 2 hours, it means that I need to pay attention for the first 2 hours significantly. We still pay attention for the entire process until 5 half-lives later it's cleared. But it's that point in that point that we just look at and think, well, that's really important information. So obviously that's an important process to include. The other part that's as a reviewer as well that's really important is the risk-benefit profile. The way that I think about this is, what is it that makes your technology, your drug, your device better than what's currently available in the market? So it's more of a market question, but it's also from an investigator, reviewer, and also a healthcare perspective. We only have limited resources. What is it about your technology that justifies its use in this population, or even to the exposure in healthy volunteers? Is it worth that risk? As we know, all trials, even safe ones, have their own inherent risks. So the risk-benefit profile is a very important component. It's also a very important component from a marketing perspective, something that you need to be also aware of when you talk to investors, and it also forms your regulatory labeling and also your drug reimbursement. So to me, that's a really important area. Then there's the safety pharmacology, which Jürgen's touched on. I won't go into much more detail than just to say that anything safety related has to be GLP. And anything that's unrelated to safety, non-GLP. And that's often causes a bit of confusion with some of the drug proposals that we see. And toxicology, Gabriel's touched on this. And from my perspective, toxicology is where I spend most of my time looking as a reviewer. I find that even if someone else has calculated the dose, I run my own calculations, because it gives me the ability just to have a sense of the dose calculations reasonable, the adverse event, NOEL's appropriate, and it also just gives a better sense of what the potential toxicity is for this particular compound. And also as an investigator, it gives you a bit of insight as to what to expect and also what to explain to the patient. So toxicology is extremely important. And again, rationalization of toxicology is important. So you don't want to skip any important steps, but at the same time, you don't want to overdo it. And when you're picking just one species for biologicals, for example, it's important to pick the most appropriate model and give a reason as to why that's the most appropriate model. Reproduction toxicology is something that's often asked. Look, usually you can sneak that into the rat tox, and then you have an indication of some toxicity effects there, and essentially that's enough for first in human. And then there's they're the main components. Actually, we might talk a bit more in a few slides on some of these. And Gabriel's already talked about how we do our reviews, but one thing I like to just to highlight there is that when we compare it to the American system with the FDA, it's the FDA that reviews and approves. In Australia, the TGA, or our regulatory division, delegates that out to the Human Research Ethics Committee, and that is comprised of a cross-section of reviewers from community, legal, scientists, doctors, clinical pharmacologists, and toxicologists. And I think this is where the real advantage comes, because most of the people on these committees are very accessible. Next slide, Gabriel. Yeah, so I thought I would highlight, next one, over the last 12 months, what are the more common mistakes? And some of these mistakes are quite trivial, some of them are more serious, but this is also something good to highlight that gives an example of why it's important to engage early and prevent some of these mistakes rather than correcting them after the submission, which can lead to delay. So we'll go through these just very quickly, one by one. The first one being safety, especially toxicology not being conducted GLP. We've talked about this already, and Gabriel's touched on it. So I go to the next one. This is something that I think is quite important. Even though with the IND, a full report's required with the Australian system, you don't need to provide the entire report. However, I think it's very important to still include the raw tabulated results. The reason that's important is because then it allows the reviewer to just have a quick look and assess what the toxicology is like. And if you're like me, you would then calculate your own dose levels. If that's missing, it sometimes can be seen as a red flag. So it's better just to include that in the IB or the investigator brochure and prevent that then being a question asked later on. So if that is to be included, the other question I get asked is then what should be included as part of that? So then all the pre-necropsy, at necropsy and post-necropsy data should be included and summarised. This also includes the histopathology and also the macro and the microscopic findings. They're really important. The reason they're important is when we're looking at what's adverse and what's not adverse, they indicate what potentially could be physiological or actually toxic. And very important to include that. The other thing that is a very simple mistake that can really delay the study significantly is not having a recovery period in your toxicology data. The reason that's important, let's say there's an adverse event and that event's been highlighted. We want to know then, is that event reversible? If it's not reversible, then there needs to be an explanation as to why that event is not reversible, because that's also a red flag. It also means that when we're consenting patients or participants, we need to highlight that this drug may irreversibly damage your liver. So most drugs are reversible, and so it's very important to make sure you have that component within your GLP tox design upfront. And most reputable toxicology labs will automatically include recovery periods. It's just important to make sure you have an adequate recovery period, and sometimes even doing a pilot non-GLP study just to determine when recovery is appropriate is advisable if there's a bit of doubt. The other one to talk about that's a common mistake is the interpretation of the no observed adverse effect level. This is really important, especially for small molecules and toxicity. A lot of times we're seeing summaries where there's an interpretation of an adversity without any supporting data or any supporting rationalization. So that's also considered a red flag and can essentially stop a study. And so to prevent that, by including the raw tabulated data and then making comments as to why an event is not adverse is really important, and it can often be quite simple. And again, if there's any uncertainty, another advantage of engaging early on with specialists like Avance that can provide that input. So a good example is where we're seeing adverse events such as raised white cell count, and the comment is that this is not an adverse event, so the NOEL is not at that level. If there's no associated histopathology or macro micropathology, then we're wondering, why is that not an adverse event? Is it possibly affecting a tissue or an organ? So if a finding has an organ effect, by definition, that's an adverse event. It's not physiological unless it's happening in the control group. So we often see this with highly toxic compounds such as oncology drugs, and it's a very common finding. In fact, we expect that to happen at high doses. What we would like to see then is a rationalization as to why it's adverse or not adverse, and that can at times be very difficult. And again, another reason to engage early if there's any questions about that is it's something that's quite easily fixed prior to submitting to an ethics committee. The other thing is a lack of stability studies. So I find this is important also for the clinical trial. I'll give you an example. With biological agents, monoclonal antibodies, for example, in the clinic, often the time that you're dosing a participant and the time that you actually dose a participant can be delayed for a number of reasons. It could be just intravenous access, And what happens is we have this expensive investigative drug sitting on the bench for an hour instead of 30 minutes. And often that's then discarded and then another drug needs to be used. And that can really affect the study, especially if it happens more than a few times. So to get around that, having a window of stability, whether it be just a few hours or ideally at least a day or two, that gives you that excursion stability data to say that, yes, we can leave this drug out at room temperature for at least 4 hours, rather than having it just wasted or causing a protocol deviation or something that might invalidate the trial. And that's something that's just quite easily done really early on as well. I'll just skip over some of these. So lack of certificate of analysis of potency. Look, vaccines are important and we can really accelerate vaccine development, especially now with all the changes in regulatory requirements by the FDA with COVID vaccines. However, it's still important for safety and quality control to include a couple of really common sense principles. We want to make sure that if we're testing a drug or a vaccine, that the same vaccine's being tested over time. And by missing that can affect that data and it can invalidate the whole trial, and it's a very expensive mistake that I've seen already happen on 2 vaccination trials that can be easily avoided. Oncology studies, not considering additive toxicity for combination studies. We're seeing a lot of combined drugs and even combined experimental drugs, and guidance allows us to some leniency in this area. However, at the same time, there needs to be rationalization of that additive toxicity and a demonstration in vivo. Another thing that's really quite unique to oncology is it's really important to try and get some indication of whether or not the drug's working. And you can, because the therapeutic window in oncology is quite unique, and they operate those trials in a bit of a different way, that you can sometimes get some efficacy data early on. It just has to be modeled in a way where it's not underpowered, and that has to be taken into consideration as well. Also with oncology studies, what we see with some submissions is the absence of standard of care, and that can cause a significant problem. So for example, if your drug is being given and yet there's no arm where standard of care is being offered, that's unethical and that can cause significant problems. So it's good to incorporate standard of care for all trials without that affecting the trial in a negative way. It's also good to predict upcoming standard of care. So having the end in mind as to what your drug development strategy is going to look like in 2 years' time. What will standard of care look like in 2 years' time? Is your drug still going to be competitive in 2 years' time? If not, you need to build that into that drug design early on. Starting dose can sometimes be too low in biologicals, but there's ways to overcome that. And I won't go into a lot of detail, but receptor occupancy is really a smart way of overcoming that. Another one that's coming up a lot recently is just this lack of independence and safety review committees. That can be a real red flag for first-in-human studies, especially, this is an experimental agent, and safety is the most important consideration, and it's also the primary objective. So in order to make sure that data and the escalation is appropriate, it really needs to be decided through an independent voting committee. It can't be comprised of above 50% sponsor. So the best way to overcome that and make sure that the data is validated is just to have independence, and an independent medical monitor is required for all phase 1 first-in-human trials. So I'll just finish on by just saying that seeking advice early and having input early is really important. It can save a very good trial from potentially not being approved. It can also prevent mistakes that could be costly, and also delays on having that drug developed. I've seen many good designs and good drugs not get through development because of just very simple mistakes that could be prevented. So I think the take-home message to me is just really get that input early, get someone to check it, have some I guess, regulatory advice given quite early in a strategic way, but also think in the end, with the end in mind. It's really the registration, the risk-benefit profile that you're aiming for. So that can be designed right at the very beginning of your phase 1 trial. We usually do this with oncology trials within the exploratory endpoints, where we're trying to get early indications of efficacy, whether it be biomarkers or other ways that are actually quite creative, that can then be used to help design the phase 2 and phase 3 trial components in a much more strategic, efficient way. And that's all. Thanks, Gabriel.
Thank you so much, Paul. Thank you for that. That was extremely informative. Very much appreciate your insight from an ethics perspective. So, Shannon, perhaps this is a good time for me to pass back to you. Thank you so much, and ask if there are any questions on the material that has just been presented.
Yeah, thank you very much, Gabriel, and thank you, Paul and Jürgen, for all those insights. We've had a few questions submitted from the audience, so I might just start with the first one. Jürgen, what do you see as the major advantages of biotech companies engaging ClinicReady by Avance Clinical to manage and guide their early drug development activities?
Yeah, thanks, Sharon, for the question. So, yeah, this really gets back to what both Gabriel, Paul, and myself are talking about, and that is really understanding, what the endpoint in the clinic is, and where the drug is going to really differentiate. So, really establishing a target product profile, and we can certainly help with establishing that, in the first place, and then making sure that all of the preclinical investigations that you're doing are really sort of marrying in with that and are fitting in with the key studies that you need to do to really satisfy, first-in-man studies. So really understanding, what all the kinds of pitfalls are in terms of, missing information in HREC submission, and also, just making sure, again, some of these later stage studies, particularly, IND-enabling tox studies, the expense does increase, and just making sure that, we're sort of setting these up so that they're going to deliver the right information, early.
Yeah, thank you. And what would you say Avance Clinical's model in helping clients with the conduct of their preclinical studies?
Yeah, look, I think the model is, it's a really collaborative kind of arrangement. We like to engage and see ourselves really as an experienced surrogate drug development team for clients. And, whilst we don't actually do the preclinical studies ourselves, we have a very good relationship with all of the specialized CROs. So it's more about a sort of a consulting service to, as I said, doing a gap analysis on the program, identifying what the key studies that need to be done are, and then, working with specialized, CROs that we have a good working relationship with to make sure that the right data is delivered within the time frame. So that's how we work with it.
Mm-hmm. And again, ClinicReady by Avance can help activities to the selection of a clinical candidate, but what happens next?
Yeah, so of course we can manage all of the preclinical studies up into the clinic, and then obviously into the early phase clinic. But typically, one of the advantages of engaging with Avance early is that not only can we help you to map out what the key preclinical studies are, but we can actually also use this to start populating the investigator brochure early. And the advantage of doing that, it means, is that you're actually ending up with exactly the right information that you need to then support your first-in-man studies. So typically what would happen is, we would start populating an investigator brochure, which would be required for the ethics submission in Australia. And we can also help with, helping to organise pre-IND meetings and these sorts of things. So we can help with the regulatory submission. Yeah.
Thank you. And Gabriel, for yourself, how does Avance Clinical assist their clients with getting approvals to conduct first-in-human trials in Australia?
Yeah, thanks, Shannon. So the way we see this is basically, I think both myself, Jürgen, and Paul stress the fact that clients should actually seek out advice early. So come and see us early in your development. we can advise you as to, the best way to actually build your, so-called pre-IND package, if you like, that will get traction with Australian regulatory for moving to clinical trials. And I guess for us, the best way is to be engaged as an extension of their team. I think, Jorgen, you used the term surrogate, and that is indeed a good term. And with many of our clients, we actually do work, if you like, as part of their team, giving early advice as to what should be there. But also, it all culminates in the preparation of the investigator brochure. And I gave some examples. Paul spoke a lot about presenting raw toxicology data, either in the actual raw tables and in the summary tables that I showed some examples of. That is really important. And I think a lot of we see a lot of clients come to us without that information in place. And this is really important in order to be able to sort of achieve the streamlined, fast-track start of trials, whereby you don't go to an ethics and then they come back with so many comments, some of which may even require going back and repeating preclinical studies. So we safeguard our clients against that, particularly if they engage with us early during their development.
Yeah, I think that's a really good point, actually, because I think when I worked at CMAX, often you would see early studies that were about to be submitted to ethics, and the comment was, if it was reviewed a little bit earlier, there's a lot of mistakes that could have been avoided that could have prevented significant costs. So I think that early engagement, even though it might cost a little bit up front, actually is essentially saving money, because you're saving unnecessary studies or the risk of delaying a really important timeline because you've missed something that is quite easy to address early on. And usually if you've missed it by the time you've had it reviewed, you can sometimes be asked then to do extra than what you would have been asked to do if you didn't miss it. So whenever there's a red flag, it usually means there's something, it can highlight other issues. So it's really important to try and prevent that. And I think you summarised that well when you mentioned that it is a streamlined process, as long as everything's prepared properly. And I think with small molecules, it's all box checking exercise. However, nothing's straightforward anymore. We're seeing much more complicated drugs come through where people are using a box checking exercise approach, which is not necessary. Ineffective, inefficient, expensive. Why not start off with a rationalized, efficient process that's really almost guaranteed to be successful, because Avance has got such strong network links with ethics reviewers, clinical pharmacologists, toxicologists, principal investigators, people that actually do the work, design the studies. It's very collaborative, and I think that that's a huge advantage and a competitive advantage, but ultimately it will allow sponsors to have a very much a streamlined approach that will benefit them in the long run, even though it might cost some upfront.
Yeah, thank you. And I know this is the final question that's been submitted, and I know you've touched on this a little bit already, Paul, but what is the difference between a submission to FDA and the equivalent regulatory submission requirements in Australia for first-in-human trials?
Thank you. Yes, as we've talked about, I think the streamlined approach, if everything's lined up well and all the studies that are required to be done have been conducted and that information is there. And I find that often the information is available, it's just not included because people from outside of the understanding of how Australian regulatory markets work sometimes assume that they don't need to include the raw toxicology tables. And so it's just a very innocent mistake. However, that innocent mistake can then cause a red flag at review. So I think that's probably the key message, is that where the process is really advantageous in the sense that it's streamlined, but that doesn't necessarily mean that our standards are streamlined. Our standards, if anything, are sometimes even more conservative when it comes to safety. But that should never affect the reviewer process, because that data is available, it just needs to be included and it needs to be presented in a strategic way. And I would say that that's the main advantage and the difference between the US process. It's much harder to do that in the US because it's a big department you're faced with. And so FDA is a big department and their time's limited. And so it's often good to get the pre-IND set up even here in Australia and have those types of discussions here, because that's all relevant to what will be submitted for an IND package later on anyway.
Yeah. Thank you, Paul. And I think that's all the questions that we've had submitted today. If you have any other questions, please feel free to send them through to Avance Clinical, and we'll refer to the relevant people. Thank you for listening, everyone, today, and thank you, Paul, Gabriel, and Jürgen, for your time. We really appreciate it, and thank you.